So, welcome to The Scoop, NANP's monthly educational webinar series. I am your host, Andrea Granson, Education Program Specialist for the NANP. These webinars are recorded, and everyone who registered will receive a link to the recording within the next 24 hours. NANP members can earn 1 continuing education credit for participating in Scoop webinars by taking the corresponding quiz, which can be found in the membership area of the NANP website. Today's webinar will conclude with a Q&A session, so feel free to type in your questions as we go along so that we can get to as many as possible at the end of the presentation. Okay, so let's get started. Dr. Armine's passion is putting together the healthcare puzzle. He specializes in diagnosing and treating complex multifactorial illnesses with a concentration in neuropsychiatric expressions, stroke autism, and chronic illness. Dr. Armine is one of the few healthcare practitioners worldwide specializing in correlating the genetic data with neuroendoimmunology, acquired mitochondrial dysfunction, and cell wall integrity to identify hidden imbalances. He develops individualized treatment plans specific to the history and the physiology of the individual patient. Dr. Armine lectures practitioners in the USA, UK, and Australia, as well as treating patients worldwide. He constantly researches about the latest findings in genetics and functional medicine. Welcome, Dr. Armine. Thank you so much for joining us today. I'm going to hand it over to you now.
Thank you so much. I appreciate this opportunity. Hello, everybody. This is my first time lecturing for the NANP, and I'm very honored to do so. Excuse me. I was asked to talk about the book that was authored by myself and Elizma Lambert, Leaky Gut, Leaky Cells, Leaky Brain. Elizma Lambert is a naturopath in Australia. She's a good friend of mine, and we have been working together for many years. She's known as the Oat Whisperer. If you want to know about organic acid testing, she knows everything and anything about it. She is what I like to call a specialized generalist, which is I'll get to in just a couple of seconds. As you heard my background a little bit, I also have a Bachelor of Science in Nursing. I'm also a paramedic, and I was a certified emergency nurse and a critical care nurse. And I've been a healthcare provider for about 44 years. Just some acknowledgements. I want to acknowledge the NANP for allowing me to share these concepts to help you simplify your understanding of complex concepts, and more than that, give you a way of explaining to your patients these complex concepts that are critical to their healing. Elizabeth could not be with us today and gave me permission to use, her portions of the book. Jillian Crowther, on the right-hand side here, is a fellow of BANT, and she is the chief researcher for the Association of Nutritional Medicine. And she allowed me to use her cell danger response slides. And of course, Dr. Ben Lynch, who allowed me to use some of his pathways from Dirty Genes. Okay, so why did we create this book? Well, we all know, being practitioners, that people are suffering, they know not why, okay? The true reasons for illness have been ignored or ostracized. And practitioners and lay people alike are convinced that syndromes are actually diagnoses. One of my favorite jokes to say to people is, if you have a sore throat, that's a symptom. If you have strep throat, now I know why you have it. So if you have ADD, ODD, OCD, schizophrenia, almost anything that you can look at that is a syndrome, it is often treated as an end result or an entity unto itself, when the real question should be, how did it get here? Okay, and it's not good enough just to find the root cause. We have to be looking even deeper. We're here because there's an overdependence on scientific evidence and specialists. This happened in the 1960s and 70s. So we live in a society that's dedicated to managing, not curing, chronic illness. And our patients, our clients, and I'll use patients and clients interchangeably, they don't have credible understanding. They don't have credible, understandable information. And the world needs really true clinicians, which are us. So the allopaths have forgotten some simple truths. They base their treatment solely on scientific proof as demonstrated by placebo-controlled double-blind studies. They ignore observational or anecdotal evidence that may lack sufficient scientific evidence. In other words, they can look at somebody and say, that can't happen. They ignore intuitive insight, and they forget the wisdom of Albert Einstein, who said, the intuitive mind is a sacred gift, and the rational mind is a faithful servant. And we've created a society that honors the servant and has forgotten the gift. It does fall to us to be clinicians. And any of the lectures I've done, and I've done a lot of lecturing in the UK, I'm on the stage yelling, we are clinicians, we are clinicians. Rather loudly because I'm from Brooklyn, so I can scream pretty good. And the reason I say that is because a clinician will use the science available, will use their clinical acumen, and use their intuition. Those are the best clinicians out there, and frankly, the best detectives. That's what we really are, medical detectives. So for our patients, our clients, education is mandatory and simplicity is the key. So what are we going to do today? We're going to talk about why leaky anything is a health issue. Okay, I'm going to be very simple. We could really go deeply into any particular area. And if there is a desire, I would be happy to come back and lecture again. A lot of these things, when I say my passion is chronic illness, everything that goes into it, every aspect of it is my passion. And we need to remember, not for ourselves, but for our patients, we have to reorient them to the importance of cellular function and cell membrane integrity. They need to know about the cell danger response simply. Okay, we need to know what damages cell membranes, what are the effects of mitochondrial function, what is a leaky gut anyway, what is the leaky brain, the role of genetic SNPs, because to some people there's a little overemphasis on genetic polymorphisms when they are just simply pointers. And frankly, how do you fix these things? My son, Jesse, was struck with schizophrenia when he was young. He's doing very, very well now with a combination of the various things we're talking about to let you know that even the most difficult cases can do well, have a good life. He's a graphic artist now. This is the leaky cell mandala. If you take a quick look at it, you see the cell membrane around it with the mitochondria and the plasmic reticulum and so forth. So the first concept that I want to get across to clients and in my mentoring groups is the importance of the cell itself. Everything in the body happens within the cell. Things are not outside the cell. We're not looking at, detoxification outside of a cell. We're not looking for methylation, glucuronidation, the biopterin pathway, transsulfuration. This is not happening outside the cell. It's all happening within the cell. So the cell is the structural and functional unit of the living organism. And what I tell my patients is, let's face it, if you put cells together, you get tissues. If you put tissues together, you get organs. And if you put organs together, you get a functioning body. But if your cell is not working, then you're not going to heal. And frankly, I think that when I say we've, I'm not being accusatory, but I'm kind of stating where our concentration should be. And frankly, our concentration should be supporting the cell membrane because it's the key to healing most anything. Look at the membrane and you'll see that all the receptors are on there. It's a phospholipid bilayer. It contains all the protein channels. It keeps in what's supposed to be in. It keeps out what's supposed to be out. And it lets nutrients in. It lets bad stuff out. And it is responsible for the signaling that happens in your body. Okay, so I've come up with the thought, and it isn't somewhere in the literature these days, that the master of the cell is not the nucleus, but it's the cell membrane. Again, why? Cell membrane is selectively permeable, protecting homeostasis. It allows for sending of messages throughout the body. It contains integral proteins, transmembrane proteins acting as pumps or channels, contains receptors that provide for the signaling of various biological functions, and thereby plays a key role in the immune system. So concisely, and remember, your patients are interested in the overview. They're going to be impressed that you know a lot of stuff, but as soon as it gets confusing, Okay, it's like, okay, thanks, doc. I understood about 10% of that. So what do we do? They really want to know what's wrong and what do we do. So if nutrients and toxins can't move freely in and out of the structures, they get stuck. You end up with nutrient deficiencies, toxin accumulation, expression of genetic predispositions. And I said that exactly the way it happens, not the polymorphisms or the genetic problems are not the cause of illness. Otherwise, the illness would have been there when you're born. Okay, something has to set up an oxidative stress load to express the genetic predispositions, and essentially that creates illness. And frankly, it's that simple. Okay, by the way, you ever wonder if there's a common ground to all illness? And we've all experienced this since we've been in practice. Why do some patients remain ill despite treatments where others get well? And sometimes, and I know I've experienced this, sometimes you feel like you're guessing or treating the wrong thing because the ones that you're banging your head against the wall about are the ones that are just not getting better despite your best efforts. And a good clinician, which you all are, will sit there and really try and do their absolute best to figure out what's wrong, and can be really difficult. So I think maybe the premise may be wrong. Maybe we're treating the wrong things in the wrong order or at the wrong times. A little while ago, I created a little chart. I did this for myself and I ended up using it in lectures, that many diseases have common symptoms. So what I did here was I put chronic Lyme, fibromyalgia, ME, CFS, and dysautonomia. And when I looked at the symptoms, I said, wow, they all have like a lot of symptoms that are really the same. Okay, so then I said, maybe there's a common causation. And the common causation was delineated by Robert Navio in 2013 as the cell danger response. Okay, Dr. Navio is an MD, PhD at the Metabolic and Mitochondrial Disease Center at the University of Southern California Medical School at San Diego. And he created a paper that is quite complex. And I'm going to go over some of the complexities of what happens and then show you how to explain it very, very simply. Okay, the cell danger response is the response of your cells that protect the cell and the host from harm. Okay, it's caused by encountering chemical, physical, biological threat that exceeds the capacity of the cell for homeostasis. So if you get hit with anything that exceeds your ability to compensate, this particular mechanism will kick in. Okay. And again, you could read this paper. It took me 3 weeks the first time. Good luck. But I'll try and explain exactly what happens. Okay, so when danger is detected, the mitochondria will alter their cellular metabolism to shield the cell from further injury. And it kind of happens in this order. So the first thing that's going to happen is the mitochondria will decrease oxygen consumption to oxidize the cellular environment. What it does is it creates within the cell more reactive oxygen species to kill off whatever's attacked it. It will stiffen the cell membrane to limit the pathogen's egress. It will release antiviral and antimicrobial chemicals. Now, specifically, it releases hypochlorous acid, HOCl. During the COVID crisis that's still going on, there's been a great use of hypochlorous acid out there, okay, because it is what the cells use to kill off anything that's attacked them. So you can actually use it on the skin, anywhere else, and it is antiviral, it's antimicrobial, it's antiparasitic, it's antifungal, and this is what your body actually uses. There's increase in autophagy and mitochondrial fission and mitophagy. Mitophagy, I can never pronounce that word. It's one of those things that makes my tongue twist and fall out, which basically states that any cells that are ready to die, this is going to help them break apart and get recycled. There's changes in DNA methylation, and because SAM is being directed elsewhere instead of going into its methylation duties, which when we all talk about methylation, remember that we're talking about the process that the body puts methyl groups on various genes to either turn them on or off. This interferes with that process, okay? We have enteric retroviruses that get mobilized, and warning happens with extracellular nucleotides. But that's complex, okay, and it's kind of interesting. But the fact is that if you can't explain it simply, you don't know it well enough. Or Dr. Einstein just said, if you can't explain it to a 6-year-old, you don't know it well enough. So I created a couple of little slides here that might be interesting to you. And what I show in my lectures and I talk with my patients is the things that activate the cell danger response. What happens to your cells right at the cellular level that causes illness to start up? And obviously heavy metals, benzene, plastics, heat, salt, in other words, chemical and physical, microbial issues, mold, fungi, bacteria, viruses, parasites will initiate this process. But I won't say less known, but less recognized and emphasis not put on it is all the psychological, emotional, and spiritual things that can happen to somebody: yelling, abuse, isolation, abandonment, PTSD, non-nurturing childhood, so forth and so on. And this has just as much of an effect on the cell as if you put mercury in there or you put, had biotoxin illness. And of course, classically, not with us, but with the allopaths, any psychological, emotional, or spiritual contributions have been fobbed off as, oh, well, you can't handle your life. Okay. And that's where the stigma for mental illness comes in. So, what actually happens when the cell danger response initiates is in the left hand panel. There's temporary impediments in things like cellular electron flow, oxygen consumption, cellular fluidity, vitamin availability, and metal homeostasis, which I get to in a second because this is a key point. Of course, redox, lipid dynamics, creation of proteins, bioenergetics, and protein folding and aggregation. Everything that the cell does to repair itself, to keep homeostasis, is interfered with because the cell is attempting to kill whatever has attacked it. Okay, so when the danger is passed, we have a sequence of anti-inflammatory and regenerative pathways to reverse the cell danger response and promote healing. The impediments are removed and homeostasis is restored. But here's how you can understand this, or you can explain it to a patient. When, let's say, somebody is younger and they have a strep throat, their doctor gives them antibiotics, and usually in a few days, they're feeling pretty poor. And then in a few days, they start feeling better. And then they have this point where they, quote unquote, turn a corner and they start getting better rapidly. That's when the cell danger response has been eradicated and homeostasis, the normal homeostatic or allostatic mechanisms, are allowed to express. What Dr. Navio found was in chronic cell danger response, either like Lyme disease, where it's being assaulted by the same organism, or multiple cell danger responses, the allostatic mechanisms or the interference with the allostatic mechanisms synergize, and the cell will not, if you will, reboot. So chronic or multiple cell danger response, you've got all kinds of downstream effects. The cell's homeostatic mechanisms are ineffective and you can't heal. So healing becomes impossible unless you treat the root causes and the downstream effects. Why is this important? Because classical medicine is geared towards is on the acute care model. In other words, regardless of how long you've had something, the thought pattern is if I get rid of the root cause, the body will just heal. Once it passes a certain point, the fact is that you can eradicate the root causes and the body will not heal unless you start working with those downstream effects. And that major downstream effect is the root of all evil. And yes, money is not the root of all evil, okay, is chronic inflammation. Everything we face as practitioners is inflammation. And unfortunately, the less learned practitioners will just kind of say inflammation and then go on to the next thing. This is the problem. A lot of times you'll hear about chronic inflammatory response syndrome, which is a body-wide inflammatory condition that is usually associated with biotoxin illness, but it can be caused by anything. What it is chronic cell danger response. Even Time did a thing on inflammation and what it can cause in heart attacks, cancer, Alzheimer's, and so forth. So to make it concise, what I did was show you what chronic inflammation can cause, and it's been rampant these days. Okay, cardiovascular issues, stomach issues, diabetes, metabolic disorders, adrenal fatigue, but it loves attacking the nervous system. And this is the base reason for autism, anxiety, OCD, migraines and headaches, addiction, behavioral issues, dysautonomia, neuropathies, PMS, ADD, ADHD, depression, and many, many more conditions. In fact, in the cell danger response paper, the left-hand figure here was table 1, and it was here to show you that all the degenerative diseases, all the things that cause inflammation will allow things like bipolar disease, Tourette's, PTSD, autism, heart disease, Parkinson's, cancer, chronic traumatic encephalopathy, traumatic brain injury, will not allow them to heal because of the chronic cell danger response. So when you want to be the master practitioner, when you want to get this across to your patients, we need to get them down to the cellular level and have them thinking in that area instead of treating things like mast cell activation disorder or ADD as a primary thing, or Asperger's as a primary thing, or bipolar disease as a primary thing. And I lectured the Japanese doctors in Osaka a couple of Decembers ago, and I kind of broke this down to chronic inflammation causes all these things. And the fact is that if we look at it like that, and we say to ourselves, gee, what causes chronic inflammation? And we get it right down to the cellular level, you can actually heal people that have been considered incurable. So what's the main thing that heals somebody? And that would be our ability to create energy. And when we look at the electron transport chain, cell membranes, and energy, we tend to concentrate on glycolysis and the entry into the Krebs cycle. Okay, so generally speaking, and this is not a bad thing to do. This is something that we absolutely have to do, that either fatty acids, proteins, but mainly carbohydrates. Carbohydrates will go through glycolysis, create lactic and pyruvic acid, which for the acetyl coenzyme A will go into the Krebs cycle, which I tell my patients is just a honking amount of biochemical processes that end up in the creation of the electron donors, specifically NADH and FADH2. And I always tell them, if I say something fast, please ignore it. Okay, and that goes into the mitochondria, and at the other end, you get a bunch of ATP, okay, much more than one would expect just from glycolysis. In this particular slide, you can see what is necessary to get the citric acid cycle and glycolysis to work. And this is true in any cellular area, because frankly, if you want to fix a cell, there's two major considerations. One, you've got to get vitamins and minerals into the cell, because all biochemical reactions use B1, B2, B3, B5, B6, B7, B9, B12, okay, and a smattering of the various minerals, magnesium, manganese, iron, and so forth. If you can't get that stuff into the cells, and I'm not talking about getting it into the gut or getting it into the serum, but getting it into the cells. Okay, if you can do that, the cell will start working and will start detoxifying, because that's the main reason why it's not working. And of course, there's a whole mess of toxins and so forth. When you get to the electron transport chain, why are leaky cells, which has been the subject of our book, important? Well, I am going to hopefully stop sharing this and share something else. Okay, and hold on. Okay, because God forbid I should have it up, right? It was up. Hang on a second. There it is. Hold on. I am going to share something with you that's on YouTube. Okay. A moment again. Here it is. Alrighty. This is from BioVisions at Harvard University. We're not going to watch the entire video. I'm going to go through some things that are very specific. Okay. If you want to look at this type in ETC animation in YouTube, and this will come up. It's an excellent, and I'm not going to use it. Okay. This was done, like I said, by Harvard. Okay, it starts talking about the mitochondria, okay, and its place in the world, okay, and how it creates energy. Now, this is the inner mitochondrial membrane. This is actually what it looks like, but not those colors, I think, but okay, actually what it looks like, it's a phospholipid bilayer, okay, and it moves around just like this. Now, what's important here for you to understand is that in the inner membrane, okay, where all those protons have been put in the inner membrane as it's going through the electron transport chain, there's a high level of protons in the intermembrane space, which is at the bottom here. So, you know, they're high energy, and this is what they do for a living. They just kind of hang out and pop around. But at the very end of the electron transport chain, you have ATP synthase. And I'm going to move it over. This is ATP synthase, and it's quite accurate, okay? ATP synthase actually runs just like a, you know, like a factory. And the ATP molecules coming off of these little things that they've, I don't know if they actually look like that. But here's the thing. If you don't have a proton gradient like this, very significant here and not so much there, this is going to stop working.
Dr. Jess, people are saying they can't see the video. Now I can, so I'm not sure why.
I'm not either, so let's see. Let's stop the share.
I could see it. I certainly could. So I'm wondering if we share the link. You said it was ETC animation?
Right. And I have a couple of slides that will, okay, so people can, all right. So I'll tell you what, let's go back to the lecture itself.
Okay.
All right. And what I'm going to suggest is that you guys go to this YouTube, okay, ETC animation, and you'll see what I'm talking about. But because I thought that might happen, okay, I did some screenshots, okay? And essentially, this is that phospholipid bilayer. And on the bottom, you're seeing a bunch of protons that in the inner layer of the intermembrane space of the mitochondria, which is where the enzymes throw the proteins. Now, there's a reason why it does that, okay? Because ATP synthase, the very last complex, uses those protons to actually run the ATP synthase. It's like a mill, okay? And if you have enough protons, it will work. But if you have a quote unquote leaky cell where those high energy protons start leaking out, okay, ATP synthase will stop working or slow down significantly. You'll not produce enough ATP and the cell will start dying. Okay, so if we don't pay attention to cell membrane integrity, what's going to happen is the body will not produce enough ATP. And unfortunately, all the work you've done, coenzyme Q10, riboflavin 5-phosphate, and everything else you've done to support the mitochondrial function will be all for naught if we don't pay attention to the cell membrane integrity. Okay, so we also have a section in the book on the role of genetic SNPs. And I wish I was in front of you guys to ask how many people, you know, are familiar with genetic SNPs and how many of you guys delve into utilizing them in your practice. I'm a recognized expert in it. I've been doing it for a long time. But when it concerns leaky cells, some of the things that are important that you consider are these particular polymorphisms. Now, let me tell you that the presence of a polymorphism does not make you ill. The absence of a polymorphism does not mean you're well. So if you have polymorphisms at PEMT, which is phosphatidylethylamine and methyltransferase, okay, if you have enough polymorphisms, what it does is give you a hint that that particular person's physiology may have difficulty creating phosphatidylcholine, and you may want to make sure that they can digest their proteins or make sure that they get enough fatty acids to create phosphatidylcholine or supply them with phosphatidylcholine, okay? FADS and FADS1 and 2 It may alter the metabolism again of the phosphatidylcholine and over its conversion of fatty acids to phosphatidylcholine. So when I am looking at a genetic report and I see PEMT and FADS together and I see significant polymorphisms, I say to myself, that person may have difficulty in those functions. By the way, when you see nothing next to a gene, it means that it's working the way it's supposed to. If it's heterozygous, or which is usually yellow, it means that the enzyme innately is working at 60% efficiency. Innately means no other factors involved. And if it's red or homozygous, the enzyme is working at about 20% efficiency. But understand that there are many variants. The classic thing is MTHFR, methylene tetrahydrofolate reductase. I know this is a little bit of an aside. Okay, but we've all gone a little nuts with MTHFR, which is only one part of the methylation pathway. Nevertheless, generally speaking, when you see a test, they've only considered the C677T and the 1298C variant. There are 50, count them, 50 variants of MTHFR. So if you were able to see a goodly amount of them, in one of the reports out there, you can see maybe 15, and there's only one polymorphism, and it doesn't matter where it is. Okay, how much of the reductase activity is going to be affected? Not much, okay? But if you had 12 out of the 15 polymorphic, the probability of the reductase activity being effective is much higher, which is the reason why Strategene came along from Dr. Ben Lynch, was to show you the polymorphisms within the pathway. An interesting and an important set of polymorphisms or genes is a DAO, diamine oxidase, which is known as histaminase, which breaks down histamine in the extracellular environment, and the ABP or AOC may affect how much DAO is produced. What I did here was, for your edification, show you the more advanced histamine pathway. Histamine's involved in leaky cells very significantly. So just to give you a little bit of orientation, anything that's in orange upregulates this enzyme. Anything that's in purple downregulates it. The green is the cofactor. So when histidine goes past histamine decarboxylase to create histamine, histamine is a important substance. It's used in the nervous system, a lot of different places. It goes into storage in the mast cells, basophils, platelets, neurons, gastric mucosa, and it goes into the storage in those areas. And these receptors, when they're stimulated by whatever stimulates them, okay, release appropriately. But if you look at the bottom symbols, if you see something like this, it's considered fast. And this is Dr. Ben Lynch's way of getting away from the heterozygous and homozygous. If it's purple, it's slow. And if it's multicolored, it's complicated, which means you have to read about it. Generally speaking, what will slow the release of histamine are things like theanine, curcumin, urtica, quercetin, rutin, astragalus, vitamin C, things that increase the release of histamine, lots of dopamine, epinephrine, norepinephrine, estradiol, but mainly you're going to see always inflammation, lipopolysaccharides, which are things that come from various bacteria, infections, allergies, which is from leaky gut, okay, trauma, shock, acetaldehyde, which comes from candida, alcohol. So if you go to the particular pathways, the extracellular pathway uses diamine oxidase to break the histamine into an aldehyde, and then the aldehyde dehydrogenase turns it into acetic acid. You're going to see this a lot around the body. Okay, the cofactors here are copper, calcium, and water. This reaction produces ammonia and hydrogen peroxide. And in me, this is mine, okay, this is said to run slowly. Okay, and worse, if I look at my aldehyde dehydrogenase, no matter where I go, it's going to be working slowly. What can I do about it? Okay, first off, make sure I don't have a lot of candida, because that produces acetaldehyde. And you'll know that because if you drink alcohol, which I don't, okay, you will get a hangover very fast because you can't convert the aldehyde to the acetic acid. But zinc and B3 are the things you need. Okay, what slows this down will be infection, reactive nitrogen species, aspirin, oxidized fatty acids, in other words, inflammation. In the intracellular pathway, histamine will be broken down by HNMT, okay, which needs SAMe as a cofactor. It'll create methylhistamine. And then this is where MAO-A and B come in. And MAO-A and B, you usually see in biopterin and how to break down catecholamines, but you're going to find it a lot of places in the body. Again, the histamine is broken down to an aldehyde. Aldehyde has to be broken down to acetic acid. The last thing you want are aldehydes running around. Doesn't matter what the big long name in front of it is, just think of it as formaldehyde, because that's essentially what it's going to do. So for myself, I'm looking at fast acting MAO-A and B, which means I should be able to, get rid of this stuff rather quickly. But things that slow it down, smoking, caffeine, being male, acute high stress, okay, things that you need, B2, okay, things that'll quicken it right here. But notice in the pathway, you need B2, B3, B5, okay? If you can get the B vitamins and minerals into somebody's cells, you're going to be pretty good, because even if you can't go through all of this, that's a real basic thing that we're trying to get across in the book, okay? Here it is again, that's histamine and methyltransferase, HNMT, your MAO-A and Bs, and things that are also involved in cell membrane integrity are your acetyltransferases. There's a lot of arguments about the FUT2, the FUT2. Okay, it has to mainly do with your ability to conjugate B12, but there is a secretor and non-secretor type. Okay, that frankly I think too much emphasis has been put on it, because what can you do about it except support the enzymatic pathway? And just for your garbage pail of knowledge, for your edification, if you see HLA-DQ2 or HLA-DQ8, okay, that's a higher incidence of celiac disease. Okay, so basically, I was leading to in this book, and for you guys, going towards leaky gut. Leaky gut, are you guys still there? Okay, yeah. Leaky gut is more of a euphemistic term, okay? Think of it as hyperpermeability, the ability of the antigens to get through the basement membrane, okay, and create an upregulated immune system. But the expression of that is all these different conditions. So when somebody has any chronic condition whatsoever, consider leaky gut. And frankly, don't be overly dependent on the tests that are out there. I've seen tons of people with what appear to be normal tests have nasty leaky gut syndrome. How do I know that? Because when I treat it, they get better. By the way, when we get to treatment, understand something. If someone argues with you about the presence of leaky gut because all the tests are negative, okay, think about this. We know that leaky gut can cause, okay, all these nasty things which I'm about to go through, okay? What's the treatment for it? Well, we'll go through that, but the treatment is fairly benign. Okay, so what's the risk-benefit factor? If there's no risk and a high probability of benefit to that person, are you hurting them? No, okay? But if the leaky gut gets ignored and not treated, what's the risk there? That inflammation will continue indefinitely. So things that damage the GI cells are the same things that damage any cell, okay? Bacterial endotoxins, medications, alcohol, inflammatory foods, chemicals, lack of fiber, lack of short-chain fatty acids that are created by friendly bacteria, and thus the lack of friendly bacteria, okay, you don't have enough butyrates, okay, or short-chain fatty acids lead you to butyrates. There's two things that happen. One, you may have actual holes where antigens get through, but then you have surface reactions where the mucosa swells and there's less of an area for absorption of nutrients and minerals and so forth. So to be overly scientific, this is what happens in leaky gut. Okay, whatever stressors occur allow white blood cells really to start attacking the cells. Okay, one, where's 2 and 3? I don't know. All right, anyway, so it attacks the cells and these cells begin to die and release cytokines, which bring in white blood cells, okay, and then release a lot of enzyme destroying stuff, destroying substances that open up the tight junctions. When the tight junctions get opened up, antigens get through, and they are chewed upon by macrophages. And that macrophage is now called an antigen presenting cell. And I tell my patients, sort of like a maitre d'. Okay, that antigen presenting cell will go to a naive T cell, which under cytokine control will determine what type of cell it will create, either a B cell, in other words, either a killer cell or an antibody. Okay, now here's the thing. That whole process initially takes about a month to happen. As the process goes on, the body can't afford to have that whole process happen and wait for a month for a response. So it creates memory cells, whether memory B cells, memory T cells, they both exist. So when that antigen comes through, you get a more immediate antigen antibody reaction. That's supposed to be protective, and it is, except when it gets a little bit too much. If we allow leaky gut to continue, what will happen is you develop a massive amount of memory cells. So even a little bit of an antigen comes through, you get this massive reaction. And the more immune upregulation you have, the more damage is created. To put it simply, GI inflammation happens from things like stress, toxins, diet, drugs, pathogens, organ malfunction, pH, candida. Okay, the first thing you're going to begin to see are food intolerances. Then you're going to start seeing immune dysregulation. Then you'll start seeing autoimmunity. If you take a really good history, okay, you'll see this particular progression happen. Okay, in the brain, it may be depression, anxiety, ADD, so forth, skin, thyroid, which is spelled wrong. Okay, the colon, sinuses, joints, adrenals. Guess what? This amount of inflammation caused by the leaky gut, which is downstream of whatever root causes there are, affect the whole body. And if you don't treat the leaky gut and you get rid of the root causes, the inflammation continues. I'll give you an example. In the 1960s, yeah, I'm an old guy. I'm like 65. In the 1960s, all you saw people doing was eating meat for breakfast, meat for lunch, meat for dinner. That caused a lot of problems like colon cancer and so forth. So there was macrobiotics introduced. Okay, that was grain. That was really good at first. Okay, people were getting better. People who wrote the books made millions of dollars. Okay, but what happened after that? We started getting reactive to the grains. And yes, the GMO gluten, which was considered high protein gluten, 400 more times than normal, okay, was getting into the system, and the body started reacting to it because it was getting through the leaky gut. So what do we do then? We put people from gluten to soy. Found out in a little while they got allergic to soy, okay, or reacted to soy. Okay, casein got in there with dairy. Okay, and we all know the big reasons for that, but to make it really, really simple. And these days, when you put somebody on a presumptive diet, you're kind of pulling out gluten, casein, soy, and corn. So what's the big dietary thing these days? Paleo, which basically goes back to meat. I know it's different, or keto. Why does this happen? Why are we having to switch our diets around? Because the presence of leaky gut has been downgraded. If you fix the gut, the antigens can't get through. Then all you have to do is work on digestion. But the common wisdom has been, okay, let's just take them off this food and that food. We do food allergy tests. And those food allergy tests will show us what the person's reacting to. Okay, great. We'll take them off that food, put them on something else. 6 months later, you redo the test, and the things that you were concerned about are down, but now you have other problems. It's a typical leaky gut situation, okay? So how do you fix this? Well, you have to fix the cellular physiology. That's the primary thing that we're trying to get across. So absorbable multivitamins, multiminerals, okay, and I mean absorption into the cells. You can do that intravenously. Liposomally is really the best and most available. Transdermal will get it from the skin into the bloodstream, not necessarily into the cells, but if you have a bad gut, nothing's getting through. The use of digestive enzymes let's face it, everybody after age 35, unless they're in excellent health, aren't breaking down their foods real well. Okay. They need a good source of lipids or liposomal phospholipids, depending on how bad they are, and relieving the environmental burden, which we're all experts at. Repairing the gut is the most important thing because it's always involved in the creation and the continuing of chronic inflammation. There's some basic things you have to consider. Okay. And I don't want to sound like, I'm telling you your business because I know you'll know this, but my whole practice is treating those outliers, that 1 or 2% of people that have been here, there, everywhere, and just haven't gotten results. And I have to tell you that every time I ask them, gee, what have you been doing for your gut? I get things like, well, I'm taking probiotics. I'm like, well, that's not enough. Okay. Always, I'm seeing that the repair of the gut has not been complete. And whether they read about it or they saw a practitioner that didn't know the principles, I'm not really sure. But the first thing you have to consider is creating a mucus layer. Mucus layers can be created by fructooligosaccharides, galacto-oligosaccharides, xylitol-oligosaccharides, and you can also use human oligosaccharides. And there's many other things that break down into creating a mucus layer, because if you don't have mucus, your biota, your microbiome, you start throwing probiotics in, they've got no place to live. They eat the mucus, and that's when they start doing their work. You have to provide for cellular physiology and GI immune function. Butyrate you can give externally. Serum-derived bovine immunoglobulin isolates. Okay, these were researched. These are not dairy. These are from the bovine serum. And the research out there shows us that it will actually help the guts of HIV patients return to normal. Okay, things like Mega IgG 2000, SBI Protect, Mega Mucosa, those are all the serum-derived bovine immunoglobulin isolates. Tight junction support, usually people use zinc L-carnosine, and supporting a broad microbiome. There's loads of considerations for probiotics out there, but the broad microbiome can be achieved by yogurt, kefir, sauerkraut, tempeh, anything that's essentially fermented. But the research out there shows that some probiotic strains seem to be more effective in treating certain conditions. There's been research into psychobiotics. So what we did here, Elizma and I, was put a list of the various probiotics that will affect inflammation, immunity, constipation, diarrhea and inflammation, like Lactobacillus rhamnosus GG, effective in quelling anxiety and depression, Acidophilus elacea, bifidum, Bifidobacterium bifidum, longum, so forth and so on. May want to consider they do sell things like this with the gearing towards a particular condition, and it actually works. I know that Seeking Health uses probiota histamine X, which is a combination of probiotics that usually chew on histamine. Okay, so your choice of probiotics is important, but sometimes it's just important that you get a broad-based probiotic. Okay, one of the things that has gone around the world pretty well right now is gut butter. Okay, which was originally given to me by Dr. Jody Dayshore from BioNexus Health, who's an incredibly gifted practitioner. Okay, and the reason that this works really, really well, I've been treating leaky gut since way before it became a thing. And aside from digestive enzymes, it's always having to take a bunch of things. Okay, and the more you ask your patient to take, the less cooperative they're going to be. Excuse me. So this combination actually works fairly well. Extra virgin olive oil, either a quarter cup of organic salted butter or a quarter cup of coconut oil, almond oils, hemp seed oil, unpasteurized natural honey, 2 tablespoons, whatever probiotics you're going to use, 5 capsules. This is all going to go into a blender. The recommendation is a delactate-free probiotic, but things like probiotic histamine X, and I've used others pretty well. 4 capsules of zinc carnosine, Sialex, which is sialic acid, which is going to be used for the mucus layer. Uh, Butyrate is important. If you can get the liquid butyrate from the sunbutyrate from pure encapsulations, go ahead and add it in. If not, I have my patients take one capsule of it with each dose. I put L-glutamine here because you often see L-glutamine in a lot of gut healing formulas. I tend to omit it because many of my patients have anxiety, OCD, and so forth. Generally speaking, they have poor GAD enzyme, glutamate decarboxylase enzyme function, which means that their glutamates are going to be high, which is incredibly excitotoxic, and it's not converting to GABA. So basically, you put the powders and the oil and everything into a blender, you spin it up for about 2 minutes, then put it in a jar, okay, put it in the refrigerator, give it a good stir, when it's semi-solid, and you can take a tablespoon of that a day. I have to tell you something. I've tried many, many different ways of fixing a gut. When people started using this, you would see results month and a half. We used to take 3, 4, 5, 6 months. Okay, this is a great formula. You're welcome to use it. It's not a secret. Okay, one of my patients actually made gut buttered gummies. He showed it to me last week. I'm like, give me the formula. I'll publish it. This stuff actually works. Leaky brain. Okay, the same principles. The things that create leaky gut will create leaky brain. And what does that mean? It means that the blood-brain barrier has these little punctate holes, okay, where viruses can get in. But the things you want to look for heavily when you're considering root causes is look for biotoxins and parasites, which is a whole nother lecture, and it's a worthy lecture because when you're looking at neuropsychiatric expressions, those are the things that are usually ignored or passed over, especially parasites. We tend to look at parasites only in the stool. I do a lot of live blood cell analysis. You can see them there very easily, but the hint you should always get is that if somebody's neuropsychiatric problems are cyclical and they're based on a lunar cycle, whether it's full moon or new moon, okay, you can track it because believe me, especially if you're dealing with a kid, mother's got a nice, record that they keep that you can access, then you should be thinking about parasites. Don't ask me why they get worse around those times, but they do. Okay, what is the treatment? Basically the same as the leaky gut with an emphasis on neurotransmitter balancing. Remember, when you're treating leaky gut, you're fixing the cells everywhere. I don't know how much time we got, but here we go. I'm at an hour.
We've got a couple of minutes. We've got 5 minutes. Yeah.
Okay. Well, then I'll talk very fast. Okay. But this is a chart that I created. One of my nicknames is the neurotransmitter whisperer, and this is a very cool chart where you can look at the prefrontal cortex. I'll just go over one thing here, and you can see what the functions are of the prefrontal cortex, and they are attention, judgment, impulse control, critical thinking. What dysfunction is occurring there: distractibility, impulsivity, poor judgment, laziness, tardiness, everything you call a teenager, by the way. All right, and what neurotransmitters are involved in this area. And when you listen to somebody's history, okay, you can kind of peruse this chart and have an idea of which neurotransmitters have to be balanced. Now, that's the purpose of this chart. I'll leave it with you, okay? So the other thing we need to do, and the other thing that was in this book, is determining the root causes, okay? And the first and foremost way of doing this is to listen. Okay, Sir William Osler, who's the founding father of Johns Hopkins, in 1895 said, listen to your patient, he's telling you the diagnosis. In allopathic medicine, they don't have the time to listen. We must take the time. Second, have your patient create a timeline. This way you can look for temporal relationships to various symptoms. Okay, then look at any laboratory findings. And once you've filled up your brain with all that stuff, and this is for patients and for us, we need to be quiet and listen to our intuitive insight. When your research, clinical acumen, and your intuitive insight agree, guess what? You've arrived. Okay, and always remember, guys, you were always told, or I'm always told, that can't be done. Okay, and that's the person who says it can't be done should not interrupt the person doing it. Okay, so yeah, see, I actually talk fast. So I want to thank you. Thank you for this opportunity. I do consult with healthcare providers and patients worldwide, so does Elizma. And I do run private and group mentoring. I have courses that bring people up to a high level of knowledge and function in functional medicine, and teach them how to consider and treat complex multifactorial illnesses. So I think that's that. So I will take questions at this time. Thank you so
Oh,
much.
I was going to say that was fantastic. Yeah, we're just asking, can we have the reference or citation for the brain wall? Brooke Penny is interested in.
Yeah, the last slide has a bunch of references. My apologies.
Yeah,
And yes, I can give you the references for the brain wall, but it's in those last it's in that last slide that I didn't show you. That has the it says evidence-based references.
Okay, fantastic. And is it okay to share the slide presentation when the
absolutely,
when the
please.
recording goes out tomorrow? Okay, fantastic.
Like I said, this is this is not a secret. I want people to understand. I want to give you the ability to understand how to, for yourselves, yes, but also different ways of explaining it to your patients. Because once they get the gestalt, once they go, oh, okay, all of a sudden they start healing like crazy.
Yeah, this is great. I have a quick question. You mentioned some people can't tolerate glutamate down to their SNPs. Do you recommend MSM for gut healing for those clients?
Well, I've found that you don't really need the glutamine if you do everything else, the butyrate, all the other parameters that are there, it works just fine. Glutamine is not it's thought of as being very necessary, but it's not. And you can use MSM. There's a whole mess of ways of helping the gut heal. But I do know that when I start hearing about somebody using different products and I look at the product and it's got 4 grams or 7 grams of glutamine, I know what's going on.
Okay, fantastic. Well, we love the body butter. I'm sure people are going to be keen to go and check that out. No, this has been absolutely fantastic. I want to thank you so much. I know you have a mentorship group. I would like, with your permission, just to send details with that tomorrow with the webinar, just so that everybody who registered can just learn more and perhaps reach out to you if they want to get in contact. So I want to thank everybody for joining us today. This does bring us to the end of the presentation. For anyone joining the NANP conference, I look forward to seeing you online. That's going to be Friday, April the 8th, so not long to go now. For next month's presentation, it will be Normal Bone Density Scores Aren't Good Enough by Reed Davis. So please mark your calendars for Thursday, April the 15th. Until then, have a great rest of your day. Bye for now, and thank you, Dr. Jess.
Thank you.
Okay, bye-bye.